Greetings and welcome to my channel. Today we are diving into the essential concepts of accuracy and precision. Whether you are a student or simply curious, grasping these principles is vital for interpreting data accurately. So let's embark on this enlightening journey together. So first important term related to this lecture is accuracy. Accuracy is defined as the degree of agreement between a measured value and a standard or known value. It generally refers to how close a measured value is to the true or known value. It is a measure of correctness. Let's understand this with a simple example. Suppose in a lab, any chemist obtains a weight measurement of 5 kg for a given substance, but the actual or known weight is 6 kg. Since the measured value 5 kg is different from the actual value 6 kg, the measurement is not accurate. Here, the degree of inaccuracy is the difference between the measured value and the known value, which is in this case is 1 kg. An important point to notice here is that the validation of an analytical method depends on the accuracy of that method. For any analytical method to be validated, it must consistently provide measurements that are close to the true or known values. If a method is accurate, it means that it can correctly measure the actual values of the substances being tested. In summary, accuracy is crucial in validating analytical methods because it confirms that the method provides correct and reliable measurements, which is essential for scientific experiments and analysis. One another important term related to the accuracy of the method is precision. Precision refers to the degree of agreement between two or more measurements with each other. It represents how closely multiple measurements of the same item agree with each other. Precision is generally about the consistency of measurements, not their correctness relative to the known value. Now let's understand precision with an example. Imagine a chemist who is conducting an experiment to see how many grams of a product are obtained from a given chemical reaction. Based on the established data, chemists should get 7.4 gram of the product. Here 7.4 gram is the theoretical yield of the chemical reaction. Theoretical yield is the maximum amount of product that can be formed in a chemical reaction based on the amount of limiting reactant. After conducting experiments 5 times, he receives yield of 5.3 gram, 5.3 gram, 5.2 gram, 5.2 gram, and 5.1 gram respectively. In this example, the measurements are close to each other, indicating high precision because the values are consistently around 5.1 to 5.3 gram. However, these measurements are not close to the actual value, which is 7.4 gram, indicating they are not accurate. To summarize this, we can say the chemist's measurements are precise because the values obtained are very close to each other. But they are not accurate because they are far from the actual or known value. Let's understand accuracy and precision with the following diagram. Observe the black dots in the first target. All the black dots are scattered over the entire target. So there is low precision and low accuracy. In the second target, black dots are at one place but very far away from the midpoint. So there is low accuracy and high precision. In the third target, most of the black dots are on the midpoint, but all the dots are not at a specific point. So there is high accuracy and low precision. All the black dots in the fourth target are pointing over the target's center, signifying high accuracy and high precision. Let's visualize the scenario to better understand the relationship between precision and accuracy using the example of hitting a bullseye on a target. In the following picture, precision and accuracy are visualized using blue and red darts. In the given diagram, all the blue darts are clustered together, but they are far from the bullseye. This shows high precision because the darts are close to each other, but low accuracy because they are not close to the bullseye. Here, all the red darts are clustered closely together and they are also centered around the bullseye. This shows both high precision because the darts are close to each other and high accuracy because they are close to the bullseye. There are two important components of precision: repeatability and reproducibility. Both are the measures of consistency of results under different conditions. Repeatability. Repeatability refers to the degree of agreement between successive measurements of the same variable carried out under the same conditions. So when an experiment is repeated over a short period of time with the same operator and the same instrument, the same result or acceptable variation is called repeatability. For repeatability, there are few mandatory conditions such as use of the same experimental tools. Using the same experimental tools ensures that measurements and results are not influenced by variations in equipment. Different tools, even of the same type, can have slight differences due to calibration, wear and tear, or manufacturing inconsistencies. The same operator should be conducting the experiment. Having the same operator conducting the experiment is crucial for repeatability. Different operators might have slight variations in their techniques, interpretations, and handling of equipment. These variations can introduce inconsistencies and affect the results. By ensuring the same operator performs the experiment each time, you minimize these variables and enhance the consistency and reliability of the results. The same instrument or measuring device should be used. Using the same instrument or measuring device ensures that the readings and measurements are consistent. Different devices, even of the same make and model, can have slight calibration differences which might affect their accuracy. By using the same instrument, you eliminate the source of variability, leading to more reliable and repeatable results. Environmental conditions such as temperature, humidity, etc., are kept constant. Environmental conditions can significantly impact experimental outcomes, especially in sensitive analytical procedures. Factors such as temperature, humidity, light, and air pressure can influence the behavior of materials and the functioning of equipment. Keeping these conditions constant ensures that the experiment is not affected by external environmental fluctuations, thereby enhancing repeatability. Conducting the experiment in the same location helps control for environmental variables that might differ from one place to another. Different locations might have varying levels of contamination, electromagnetic interference, or even slight differences in atmospheric conditions. Performing the experiment in the same place ensures a stable environment and reduces potential source of error. The experiment should be repeated within a short time frame. Repeating the experiment within a short time frame helps minimize the effects of time-related variables. Over time, materials can degrade, instrument can drift out of calibration, and even the skills or techniques of the operator can change. By conducting repeats in short period, you reduce these time-related factors, maintaining the consistency and reliability of the results. Reproducibility. Reproducibility refers to the extent to which consistent results are obtained when an experiment is repeated under different conditions. This broader definition acknowledges that scientific findings should remain robust and reliable even when variations occur. These variations can include different operators, equipments, laboratories, and times. Reproducibility ensures that the results are not just a product of specific circumstances but are universally valid. For example, if different chemists in different laboratories measure the mass of the same substance using different balances and achieve similar results, this demonstrates reproducibility. Thank you for watching this video. If you liked this video, kindly subscribe and don't forget to hit the bell icon for upcoming future updates.